All right, welcome everyone. It's my distinct pleasure to introduce George Davey Smith. I think probably one of the world's foremost epidemiologists, and I guess the former noise terrorism outfit member for whom commercial and artistic success eluded him. George has been, I think, one of the leading lights of using genetic and genomic technologies to bring another lens of understanding into observational epidemiology. I think the kind of impact of Mendelian randomization is difficult to overstate, but the conclusions of it, I think, if at least heeded, certainly would have saved a number of pharmaceutical companies billions of dollars of wasted investment had they been paying attention in the first place. I think George is one of the most entertaining and lively speakers I've had the pleasure of witnessing speak, and it is my great honor to introduce George to deliver a sort of 20 years on lecture about Mendelian randomization, ideas around human genetics and how they can be used to inform on questions of epidemiology. We'll spend about an hour. There'll be time for Q&A at the end, and then after the seminar, we'll have a little celebratory toast in the atrium to, I guess, cap this stunning achievement of science as it pertains to epidemiology. So take it away, George.
So yeah, thanks very much for the invitation to talk about Mendelian randomization 20 years on. I'll start with my conflict of interest statement. I've received grant income over many years for Mendelian randomization research. I direct a medical research council unit, the MRC Integrative Epidemiology Unit, which conducts a lot of Mendelian randomization research. My Twitter handle is MendelRandom. And with Shah Ibrahim, I wrote the first extended exposition of Mendelian randomization in 2003, and my cognitive conflicts of interest around Mendelian randomization are vast. So I might not be the most level presenter on this. But I'm going to go back to before Mendelian randomization started to be applied to say, you know, why develop a method like Mendelian randomization, which uses genetic variants to understand phenotypic causality? I mean, why not just directly study the phenotypes, like, you know, taking vitamin supplements, or you know, or vitamin levels, or, you know, lipid levels, or your sausage intake? You know, just measure those things directly. Why involve genetics? And the reason is that epidemiology in the last part of the 20th century came up with some rather dismal predictions or projections. So, for example, here's a couple of studies published back-to-back in the New England Journal of Medicine about use of vitamin E supplements and coronary heart disease. And people who took vitamin E supplements, this was the Nurses' Health Study, had about 35% lower coronary heart disease incidence than people who didn't take the supplements. And everything was adjusted for, statistical adjustment was made for everything that was measured. And the same was seen in the physicians, in the health professionals follow-up study. The New York Times gave the headline that vitamin E greatly reduces risk of heart disease, so pretty obvious causal claim. Use of supplements that contained vitamin E increased to about 35% of US adults. This was very consequential research. And of course, as everyone knows, most people now know, a whole host of studies which actually randomized people to vitamin E supplements, followed them up for up to 10 years, showed absolutely no effect of taking the supplements. And this was, the observational exposure was precisely the same as tested in the randomized controlled trial. It was, you know, it was actually, you know, taking the bottle, putting the tablet into your hand and swallowing it. That was taking vitamin E supplements, was the observational exposure, and that was what was tested in the trials. And Shah Ibrahim and I edited the International Journal of Epidemiology for about 16 years, and the first editorial we published was titled Epidemiology, Is It Time to Call It a Day? And I think it was the first time we used this cartoon, which I guess most of you will now have seen, about, you know, today's random medical news from the New England Journal of panic-inducing gobbledygook. And the presenter presses the button and the thing spins round and it says, "Coffee causes depression in twins." That's today's news headline. and then he presses it, and then, you know, tomorrow stress causes breast cancer in twins or whatever, or overweight smokers. No, overweight smokers are always to blame. And every one of these combinations is spurious, except red wine does cause sexual dysfunction in men aged 25 to 40. But it's an incredibly long time ago that I can base claims that that might be true. So the perception of epidemiology was certainly suffering from these many studies, many cases: hormone replacement therapy, vitamin C, beta carotene, selenium for prostate cancer, etc., prevention, etc. Many examples where observational data were not matched, or were not matched by randomized controlled trials. So we said that, you know, we discussed genes and how in 2001 and how the incorporation might help. In another field, back in 1991, I'd been working on cardiovascular epidemiology and on the question of HDL cholesterol and coronary heart disease. HDL cholesterol was already by then being called good cholesterol, and methods had started to be developed whereby you could actually elevate HDL cholesterol. But it related very strongly to triglyceride levels, and HDL cholesterol was protective. So the lower risk of coronary heart disease with higher HDL cholesterol, and triglycerides had adverse prediction. They predicted higher risk. And Andy Phillips and I analysed some data from the British Regional Heart Study. We found the usual result. This is just putting them both in the same direction to make the graphs easier. But we knew that triglycerides were vastly more variable than was HDL cholesterol. There'd been a sort of study giving a full English breakfast to people, and the HDL cholesterol hardly changed. The triglycerides just zoomed up and came down again. So we modeled different levels of measurement in precision, of measurement error in both the triglycerides and HDL cholesterol. And of course, if you model measurement error and you correct for measurement error, the predicted effect size gets greater. So it got greater for both triglycerides and HDL cholesterol. This is the red bars. And then when you mutually adjust, you see the same results. When you say that the measurement in precision in triglycerides was with an intra-class correlation, just means measuring the same thing twice of 0.7 and HDL of 0.9. But if you change that to 0.6 and 0.9, then the results flip. The HDL, the triglyceride results became more stable. The HDL cholesterol result attenuated. It looked as though triglycerides were more important. And then if you changed the 0.9 to 0.95, it flipped again. So we thought that this with the levels of measurement error that must exist in exposures and confounders, this suggested there may create real problems in trying to adjust for confounding factors. And actually, this work led to me giving up my PhD. I was going to do a PhD on job stress and how fibrinogen-mediated job stress affects on coronary heart disease. And we did this sort of modelling and realised it was impossible to ever get a sensible answer. So I'm a complete fraud. I don't have a PhD. Probably one of the few people ever to speak here without a PhD, but that's how it is. Years later, this is a massive combined analysis of triglyceride and HDL cholesterol analysis, and you see that adjusting the triglycerides led to great attenuation almost to the null. Adjusting the HDL cholesterol hardly had an effect, and even in 2009, when the first negative trial had appeared, the authors concluded that the current findings suggest that therapy directed at HDL cholesterol, as well as non-HDL cholesterol, may generate substantial additional benefit. So the issue was that the assumptions of conventional observational studies are to actually be able to interpret your regression results in any causal way is that you have no unmeasured confounding and that there's no measurement error. But the problem is that no unmeasured confounding and no measurement error means you're not dealing with epidemiological data. So the assumptions will never be met. In 2001, Nature had a little article about how epidemiology was set to get fast-track treatment by being taken over by geneticists. The VP for Novo Nordisk was sort of leading this push. But this certainly was a point, was that classic observational studies were not getting the correct answer. Now, thinking about, let's say in our 2001 editorial, we'd said about potentially incorporating genetics in studies that might help. But my sort of knowledge of genetics was, natural selection. This is just something taken off Wiki, the very standard story, but that, of course, natural selection is through phenotype. It's the phenotype that allows greater reproductive success, for example, and that phenotype drags the genes along with it. So, you might have a genetic influence on the peacock tails, which leads to the peacock ending up with more eggs from the peahens. So now, of course, Darwinism, which had introduced this notion of natural selection, had no mechanism for this, which was one of the reasons of what the period was called the eclipse of Darwinism. The engineer Fleming Jenkin had criticised the fact that Darwin's theory of pangenes would lead to blending inheritance, and no models of blending inheritance would actually allow the selection to actually move to any sort of permanent change. But of course, in Moravia at the same sort of time, Gregor Mendel had done his classic P experiments, and by 1905, 1906, Robert Heath Locke had put forward Mendel's first and second laws. Mendel's two laws do not appear in his lectures, his two lectures that became his paper. I'm always paranoid about citations, and just before the MR paper appeared, I went back and I thought I'll just check the translation of Mendel's paper to find these two laws. They're not in there at all, or there's something slightly similar to the second law, but there's about 10 things that could be called generalizations that he made. It's an absolutely brilliant paper. But the laws that we all learned were first introduced as the first and second law by R.H. Locke, the complete segregation of the two allelomorphs in equal numbers of the germ cells of a heterozygote. That's the first and most important section. And the second part is the fact that, as a general rule, separate pairs of allelomorphs segregate quite independently. But Locke already knew that there was linkage disequilibrium, what we would now call that some things did not segregate independently. There was linkage. Although that term wasn't used. Interestingly, T.H. Morgan is usually said to be the person who labelled these and introduced them as the two laws about 10 years after Locke, but that wasn't the case. Which means the next character, of course, would be R.A. Fisher, but we're not going to talk about Fisher because he's been cancelled, so we'll move on to Mendelian randomization. So one of these studies that Shah Ibrahim and I were both studying observationally was homocysteine, which was a circulating agent which was thought to increase the risk of coronary heart disease, and it was known that folate reduced homocysteine levels. Many studies appeared, observational studies appeared and did adjustments, etc., said that it was an independent risk factor. We did a study in took the sort of sensitivity analysis type view I've said, thinking forward and thinking that any residual effects were well within the range of what you'd expect from the very substantial confounding you had of homocysteine with many other factors. And published an observational study in 2001. And folate enthusiasts wrote in and criticised our paper. And there we wrote that the best evidence regarding homocysteine and CHD risk would come from studies of genetic polymorphisms, the thermal label variant of the MTHFR gene. The carriers of that have homocysteine levels that are higher than carriers of other alleles. and that if this was a causal effect, you would expect that at 5 millimoles litre higher level of homocysteine, there'd be a relative risk of 1.22. Now, this was the first MR or effect estimate, which would now be called an instrumental variable effect estimate, that I calculated. I didn't think of MR as instrumental variables, and I didn't know the Wald estimator, so I derived it myself, and I got the standard errors were wrong. So you can redo this, and you can write a letter in and say that this estimate is incorrect, which it is. The standard errors should be somewhat wider. But we then said that through Mendelian randomization, this tests the unconfounded association between homocysteine and CHD risk, and we went on to say there could be problems like population stratification or being in linkage disequilibrium with other loci influencing disease risk, and conventional epidemiological studies have a limited future, and resources should be put into either RCTs of homocysteine manipulation or further studies using Mendelian randomization. So that then a couple of years later, Shah and I wrote the first extended exposition of Mendelian randomization. And as on this slide, it was delivered as the 30th Thomas Francis Jr. Memorial Lecture. It says to be delivered, because that was the case. I hadn't delivered it at the School of University of Michigan School of Public Health on the 6th of March 2003. And I can honestly say that that talk went down like a lead balloon. People, you know, there wasn't certainly no champagne reception. In fact, at the drinks afterwards, people would come up and say, "Oh, we preferred your earlier stuff. Can't you do your earlier stuff next time?" So, yeah, so that was the first presentation. But it was, but the MTHFR example, because that was one of the very few polymorphisms that sort of existed and had been genotyped in you know, even in populations of 1,000 or whatever, was used for much of the examples. And here we just showed that the genotype, and it's tiny sample size, that the genotype wasn't related to confounders, but the plasma homocysteine was related to confounders. And we did, probably did a misspecified calculation again, but we said that Mendelian randomization allowed genetic epidemiology to make direct contributions to understanding of environmental determinants of disease beyond the essentially nominal incantation of the term environment, as in gene-environment interaction. When investigating effects of functional polymorphisms, it's possible effects will differ by environmental exposure, and there's some evidence that the association between MTHFR genotype and CHD may differ according to folate status. But however, the important message is that the association of the genotype and CHD risk does not indicate that genetic screening is merited, because of course most of the interest or the sort of projections about what genetics would contribute was about personalization of medicine, about gene-environment interactions, selecting people according to genotype for particular treatments. But we were saying the triangulation of the associations between genotype, homocysteine, and CHD risk provides robust evidence of a general causal effect of homocysteine on CHD risk, and therefore likely protective effect of folate. So between them, the much larger studies had come out, and we'd then carried out these calculations, which had tighter standard errors. But then we did 2 years later, we did a systematic review. And the systematic review came up with the conclusion, once we actually got all the data and took into account potential publication bias, etc., that in European, North American, and Australian populations, there was no evidence of the MTHFR polymorphism relating to coronary heart disease risk. And the fact that it did associate with risk in East Asian populations could be due to the fact that they were small studies and publication bias. And I think that's pretty much the evidence today. And folate trials didn't reduce, which did reduce homocysteine substantially, didn't reduce coronary heart disease. But we introduced Mendelian randomization in a strictly Mendelian way, and said that it's most clearly seen in parent-offspring designs that study the way phenotype and alleles co-segregate during transmission from parents to offspring, and in matings in which at least one parent is heterozygous at polymorphic loci, the frequency with which one of the two alleles from a heterozygous parent is transmitted to an offspring with a particular disease or phenotypic characteristic can be evaluated. So we saw Mendelian randomization as being a within family study design. because there you are actually taking the true randomness that exists in the Mendelian processes into account, and that the analogy to RCTs was in this situation where there was true randomization. You could have something which was equivalent to an intention to treat analysis. That's how we depicted MR. But at that time, there were virtually no, the amount of data from family studies was extremely limited. You'd have zero statistical power to do anything in that context. And we said that associating markers with disease risk of phenotype within populations, not within family studies, isn't as well protected against potential distorting factors as a parent-offspring comparisons, and that the Mendelian randomization in genetic association studies is approximate rather than absolute. And I think that conclusion, now of course we have evidence from large-scale sibling studies which allows evidence to be put when that is the case, that the fact that the family studies which take account of biases I'll come to work better. So you can the sort of setup we put was that in your conventional observational epidemiological studies, there's confounding and reverse causation. For example, if your modifiable exposure is homocysteine, the atherosclerotic process which develops before you have your myocardial infarction, that disease process could influence your homocysteine levels. So the direction of causation could be from developing disease to exposure rather than from exposure to disease. And the sorts of simulations we'd done, sensitivity analyses, taking measurement error and missing confounders into account, told us that it was usually impossible to exclude those things in standard observational studies. So in the Mendelian randomization approach, you take a genetic variant which relates to the modifiable exposure, in this case homocysteine, and then you can then relate that variant to the outcome, and there's no confounding of the genetic variant and outcome, and those together allow you to estimate the effect of the exposure on the outcome, and then it allowed me to do a misspecified quantitative analysis. But the fundamental assumption of Mendelian randomization, if you want to use this for understanding processes through which you could reduce the risk of disease, which is the very reason you'd want to do it, the assumption is of gene-environment equivalence. That is, that if an exposure is influenced by environmental factors like diet or smoking, then it will have the same downstream effects on your outcome as if it is influenced by genetic difference. And in Mendelian diseases, there's sort of many examples of such gene-environment equivalence. On the right here, we have pellagra, which is caused by long-term deficiency of vitamin B3 or niacin. It's a dietary deficiency. And on the left, we have Hartnupp's disease, which is a genetic disorder resulting in poor absorption of amino acids, including tryptophan, which is then converted to niacin. So they produce a similar phenotype, even though they're driven either by externalities like diet or by genetic difference. And this notion of phenocopy and genocopy is sort of widespread in developmental genetics. I mean, Zuckerkandl and Villert, in an old paper now in PNAS, said, no doubt all environmental effects can be mimicked by one or several mutations. So I think that's, I'm not sure, I think all is going a bit too far, but I think if you're thinking of an endogenous disease process, then it's reasonable. But if you're thinking like you've been hit by a truck, I don't think that can really be mimicked by a genetic variation. But but this is the principle we have to think of. Now, the interesting thing with the MTHFR was that there was this combination of folate and homocysteine, because we folate lowered homocysteine. The genetic variant was associated with lower homocysteine, but then it wasn't really known the extent to which it actually related to, for example, red blood cell folate. There's limited data. Now it's very clear that the variant relates to higher red blood cell folate, higher serum and plasma folate, and that leads to the lower to lower homocysteine. I mean, in another example, for example, LDL cholesterol, the gene-environment equivalence assumption would say that it doesn't matter whether you change your cholesterol level through eating less sausages or taking a cholesterol-lowering drug. It should have the same effect on your outcome as if that difference in cholesterol occurs because of genetic difference. And in the LDL cholesterol world, of course, we have plenty of evidence that that gene-environment equivalence does indeed apply, because the genes which are targeted by at least 4 of the LDL cholesterol lowering drugs, CETP inhibitors, Niemann-Pick C1-like-1 inhibitors such as ezetimibe, HMG co-reductase inhibitors, i.e. statins, and PCSK9 inhibitors, they target genes in which there is genetic variation that relates to a difference in cholesterol level and to a difference in coronary heart disease risk. And the fact that, of course, when you think about gene-environment equivalence, you've got to take time into account, because, of course, your genetic variation will relate to a difference in your LDL cholesterol level from birth on. When US soldiers in the Korean War who died in the Korean War were autopsied, the 19-year-olds already had plenty of atherosclerosis. The disease develops right the way across life. And you wouldn't expect that 5 years of giving people tablets that lower their cholesterol would produce all the benefit that you were estimating from the genetic studies. But here you get really rather beautiful scaling, such that the genetic studies produce about 2 and a bit times the effect size that you see in 5 years of drug trials. But the use of this is that it tells you how much of the benefit you can get within the period of time that the people are in trials. But there is a clear equivalence between the effects of the genetic difference and of environmental differences. So the limitations of Mendelian randomization we saw in 2003 were failures to establish reliable genotype, intermediate phenotype, or genotype disease associations. And this was really was one of the major problems of genetic epidemiology, was that the papers published in the Lancet and Nature Genetics everywhere, well, candidate gene studies were virtually all nonsense, because the statistical power was simply way too low, and they were just false positives selected because of analysing. You have many candidate genes to analyse. So we discussed that in detail. Potential confounding through population stratification, and potential linkage disequilibrium, pleiotropy and the multifunction of genes, and then canalization and developmental stability, which has sort of dropped out of people's consideration, but I think remains very important. And this is that your genetic differences will exist during developmental periods, and it's possible that there will be compensation during development for a genetic difference. That means that what you see from the genetic difference and later in life is not what you'd see if you produce that same difference when someone was an adult. So we could think of sort of mice, and this is one of the sort of well-known sort of knockout studies. You have your mouse, and if you think about sucking all the myoglobin out of that mouse, you would have a dead mouse, pretty obviously. But if you knock out myoglobin, remarkably, so the mice produce no myoglobin at all, you actually get live mice born. They're not going to win the mouse Olympics, but they can actually stand up, which is and walk, which is pretty amazing. So canalization clearly occurs, and I think one of we just don't consider it anymore, that the fact that tissue becomes differentially responsive because it develops in a different milieu. And it's possible that the disconnect you see sometimes between eQTLs and pQTLs, for example, could reflect that sort of process. But this is the one that really needs to be looked into. And as we said in 2003, and I think it's still true, it leaves Mendelian randomization in a somewhat unsatisfactory position of facing a potential problem that cannot currently be adequately assessed. And there was the lack of suitable polymorphisms for studying modifiable exposures of interest. Now, everything you read on Mendelian randomization these days is generally set in an instrumental framework, instrumental variables framework. We didn't mention instrumental variables because we hadn't thought of them in the MR context. I'd actually taught a course at the London, put together a course at the London School of Hygiene about ways of evaluating interventions when you couldn't do RCTs. So non-RCT ways of evaluating health interventions. And I'd looked into instrumental variables and indeed taught about instrumental variables, but they all seemed hopeless to me because it was things like the distance of people lived from a water hole. And the idea that that was actually in any way a sort of something which could tell you about how the availability of greater amounts of water would or would not influence health, water use, just seemed remarkable because it was so obviously confounded by just so many other factors. So I just sort of forgot about them. And then Duncan Thomas said to David Clayton and me, you're just talking about instrumental variables. We thought, yeah, we could have thought of it in that way. But and so I therefore invited Duncan to write this up, and he said he nicely drew the picture for us for what an IV was, and said that one could use this, think of this in an instrumental variable way. We more thought of genetic variants in the way that T.H. Morgan, who mentioned earlier, did. T.H. Morgan sort of famously had this notion that he didn't really care what a gene was. He just knew that you could use genes to actually understand biology. And in his Nobel Prize-winning speech, he said, I don't care what the physical basis of the genes are. We locate them in the chromosomes. We know they're there, but frankly, the actual idea of exactly what they are isn't of much interest to the working geneticist. You can speculate about it if you want, but there's no consensus of opinion amongst geneticists as to what they are and whether they are real or purely fictitious, because at the level at which the genetic experiments lie, it doesn't make the slightest difference whether the gene is a hypothetical unit or whether the gene is a material particle. And in some of the MR contexts, that's the case. But the instrumental variable framework has certainly been useful in terms of thinking about computation in MR. And in the instrumental variables framework, you have to have an instrument which is reliably associated with your exposure. There can't be any confounding of the exposure, of the outcome and the genetic variant. Confounding we usually think of in terms of, for example, population stratification, but another important confounding setup is that you get your genetic variants from your parents, which means that the variants you get will be overrepresented in your parents, obviously. And if those genetic variants influence the environment the parents provide for you, so if the parents have genetic variants that relate to them drinking more, you'll grow up in in a sort of wetter environment, alcohol-wise. Or if your parents smoke more, or if your parents have a higher educational attainment score, there'll be more books in the house, etc. So these dynastic effects, as they're called, could produce confounding. And we used, in 2003, the example we used to discuss this was, again, of MTHFR, because it was about the only genetic variant that was reliably associated with things. And we talked about if the mother carries the variant which is related to lower folate status and higher homocysteine, then the child has a higher risk of twofold risk of developing a neural tube defect. For the father, of course, it's an intrauterine effect. Obviously, it's a developmental effect. If the father carries that variant, it has no influence. And for the fetus, of course, because they'll overrepresent fetuses whose mothers carried the MTHFR low folate variant, they would carry that to a greater extent, but not to the large extent as the mother. Now, if you were just studying the kids, you would think that it was their genetic variant that was actually influencing the disease, but it's not their genetic variant, and it's the folate status of the mother that matters. So there are more complex forms of confounding than just population stratification. Dynastic effects need to be thought of. And then the most studied, of course, is pleiotropy, that the genetic variant can't have an effect on the outcome that isn't through the exposure. Now, an obvious thing to think of doing, and that's to test that, is to say, well, let's just adjust for the exposure. And many early Mendelian randomization studies did this, and it seems logically okay, because if you adjust for the exposure, then if the effect of the genetic variant is all through the exposure, then it should reduce the relationship of the genetic variant to the outcome to the null. But there's two obvious problems with that. Well, there's two problems with that. The first is measurement error in the exposure would leave an effect. But the second one is that if you adjust for the exposure, then you induce collider bias, you induce confounding of the genetic variant and the outcome. And an example of how conditioning on common effects introduces associations would be thinking of admission to colleges in the US, and I know some colleges will have a sporting scholarship and as well as academic scholarships. Now let's imagine the situation where in the overall population there's no relationship between sporting prowess and academic ability. So they're not associated, completely uncorrelated. But you imagine if you walked onto the campus of one of these colleges and you threw a ball to a kid in the other end of the hall, and they tried to catch it, he fell over backwards, bounced off his head. You could go up to him and say very confidently, you must be really clever, because that kid didn't get there from a sporting scholarship. So in that college setting, you generate a very strong negative correlation between academic ability and sporting prowess, whereas that doesn't exist in the population. So all the sorts of adjustments, like if you stratify for anything in Mendelian randomization, if you look just in people with a disease, that's what you're inducing. You lose the randomization. So you've got to be think very hard about the possibility of introducing collider bias in these settings. So I'll skip that. So we were very slow on the uptake about this being an instrumental variable approach. But another person introduced, both discussed instrumental variables. Indeed, he introduced instrumental variables and causal analysis, and didn't connect them in his genetics, and that's Sewell Wright. In Sewell Wright's in his biographical memoir, Jim Crow said he read his father's math books and learned to extract cube roots before entering school, a skill that he said brought him instant lasting unpopularity with the other students. Again, something some people here might recognize. And Wright, of course, introduced path analysis in this famous study of guinea pig coat color. What I love about this, and people always talk about the path analysis, people don't point out that this is his drawing. He just writes chance, and the arrow goes to the generation of the gametes. I mean, this is Mendelian randomization. This is what he was talking about with chance. It's a beautiful paper. I should skip over this, yeah, sorry, I'm going the wrong way. It's a beautiful paper, which also, incidentally, as an aside almost, introduced this notion of intangible variance, which we know in human situations are called a non-shared environment, which he pointed out, a big chunk of it will not be the sort of environment you can point to, touch or smell. It will be sort of developmental differences, purely stochastic events, etc. And Wright and his colleagues did lots of causally anchored path analyses of antenatal environment and offspring phenotypes, assortative mating, obesity. They even did lipoproteins right at the end of Wright's life. And Wright, said about path analysis that it was the Mendelian heredity which allowed you to actually anchor the analysis and make causal inference. And you could safely base this on the Mendelian nature of inheritance. And his biographer, Bill Provine, said, that Wright was rather upset by the fact that everyone just took up this path analysis and just used it in non-genetic situations. And this, of course, is in sort of structural equation models used widely in psychology and many other disciplines in a way which would actually make Wright rather unhappy, and then make causal inference from those situations. And Louis Gutman, this is one of my favorite papers, Gutman said, genetics has but one modest framework for paths. In contrast, according to current journals, sociologists keep discovering new fundamental path frameworks every month, and sociological graduate students are required routinely to hand in, as individual class exercises, new discoveries equaling Gregor Mendel's. So, path analysis is a sort of dangerous tool. But Philip Wright, Sewell Wright, and the geneticist, and his father was the economist, Philip Wright, they together developed the instrumental variables analysis notion in 1928 in the annex to Philip Wright's book. A huge amount of analysis has been done about who wrote it, etc. But the letters are available. It's clearly the two of them were writing back and forth. It really was a joint activity. So why the label Mendelian randomization? I'm afraid we have to talk very briefly about Fisher, because in 1951, and the only time Fisher ever talked to an audience both of geneticists and statisticians about statistical methods in genetics, he famously, now famously, this has hardly been cited until relatively recently, said, genetics is indeed in a peculiarly favoured condition in that Providence has shielded the geneticist from many of the difficulties of a reliably controlled comparison. The different genotypes are beautifully randomised. Generally speaking, the geneticist, even if he foolishly wanted to, couldn't introduce systematic errors into the comparison. And that he then talked about the statistics and genetics, and said a connection between the two subjects was that of the factorial method of experimentation, factorial randomization. And he said that this comes from the simultaneous inheritance of Mendelian factors. And I wrote to his daughter, Joan Fisher Box, when I reprinted Fisher's lecture, and she wrote a piece. She wrote his biography, a very good read actually, and she wrote a piece saying that this was how she saw it. So R.A. Fisher, who's seen to have introduced randomization and randomized controlled trials, the idea was from Mendelian genetics. So Mendelian randomization comes before randomized controlled trials. And but where did the name come from? When we first mentioned it, we'd got it from a paper by Graham Wheatley about bone marrow transplantation in children. And they'd used it. I'd come across this when I was doing this course on different ways of evaluating things when you couldn't do randomized trials. And it was a brilliant idea. They were thinking about kids with hematopoietic cancers. Parents would absolutely understandably not want their children to be randomized into such studies. So that we couldn't do the randomized trials, but what you could do is say, did the child have a sibling who was HLA compatible? And if they did, they were much more likely to get a transplant than if they didn't have a sibling who was compatible. So you could do an intention to treat analysis based on HLA compatibility, and they called that Mendelian randomization. It turns out, that was what I knew, because I'd found that paper. It turned out that, in fact, a similar thing had been advanced earlier, and they'd called it biological randomization, but I hadn't seen that, which is why I used the term Mendelian randomization. And others in 1989 had used the notion of meiotic randomization, and others had talked about genetic randomization, which had also been used in the hematopoietic cancer world. So we can have a vote at the end about which name would have pissed people off less. Do you think these are the possible names? Germline genotype, as telling you about specific questions of phenotypic causality, definitely existed in the field of cardiovascular epidemiology, where familial hypercholesterolemia told people that cholesterol was the likely cause. Work had been done on work had been done in the cancer field with detoxification enzymes. Martin Catan had published a letter without any data, but a letter in 1986 about apolipoprotein E, which was a genetic variant related to cholesterol, that would allow one to test whether low cholesterol increased the risk of cancer. I reprinted Martin's paper, and his letter in the IJE. Well-known example of Mendelian randomization is with C-reactive protein, where this predicts a higher risk of coronary heart disease, but it relates to every single confounder you can imagine. If you look at a cis genetic variant that relates to C-reactive protein, that is unassociated with the confounders. While the observational studies that even after you adjust showed an adverse predicted effect of CRP, the Mendelian randomization analysis suggested no effect. You can use negative controls, can be used, I think, really greatly strengthen the inference. Alcohol is an example of this. People love it on this paper saying that alcohol protects against diseases. This is for the UK papers, The Guardian, moderate drinking can lower the risk of heart attack. Independent, a bit down market. Drinking a pint of beer a day linked to reduced risk. Daily Mail, really down market. Cheers, drinkers who have one glass a night. Or the lowest, drinking alcohol slashes risk of heart problems if you drink this much per week. The rather more sober US, alcohol is good for your heart most of the time. Or the Irish Times, moderate drinking may cut the risk of heart disease. And then it says, be good for your heart. And even heavy drinking may lower your risk of heart attacks, a new study adds. This can be tested through Mendelian randomization due to the fact that in East Asian populations, people carry genetic variant that relates to the poor clearing of acetaldehyde that the metabolism of ethanol produces. And to put it in a cartoon form, acetaldehyde gives you the adverse effects of alcohol. It gives you sort of flushing, palpitations, headache, guilt. Actually, it doesn't give you guilt, but it gives you the other adverse effects. And therefore, people who carry two versions of this variant don't drink at all. People who are null homozygotes, null variant homozygous. And this was a meta-analysis we did very early on, just showing that in both men and women, the homozygous null variant folk weren't drinking. But in the heterozygous men and the homozygous wild type men were drinking really quite substantial amounts of alcohol, but the women weren't drinking in those cultures at that time, whatever their genetic variant, which gives you a really nice null zero relevance point group, because of course in the women you should see any pleiotropic effects of the genetic variants would be seen in the women. But when we did this meta-analysis, there were in women, the genetic variant didn't relate to alcohol. It didn't relate to blood pressure at all, whereas in the men, there's a very strong effect where blood pressure raised, it suggested a really substantial blood pressure raising effect of alcohol, which is bad news, especially as we're going to have a reception later. So take an ACE inhibitor before you glug the champagne. And you can use this method to estimate pleiotropy-free effects. And in a very large-scale study in China, these famous curves that get so much attention, people love these, reduced risk in moderate drinkers. The observational estimates are all sort of sadly linear. And in China, it's beautiful because you can stratify by geographical region and by sex, so endogenous variables, which actually generate groups with very, very different levels of alcohol intake. So this form of analysis allows you to get the shape of the relationship without doing any stratification on colliders, because obviously genetic variation related to drinking doesn't change your sex. So you need to get stratification variables that can't be colliders. When I lived in Glasgow, they used to drink Old Mortality. This was a beer, so I won't be able to sue them for when I had my heart attack, saying that they were telling me what was going to happen. But around here, very sadly, there won't be a randomized controlled trial of alcohol and cardiovascular disease, as the NIH shelved the trial because of the links between the people who are going to run the trial and the alcohol industry. That's a very sad story. Multivariable Mendelian randomization is very powerful because it allows you to do multivariable analysis, which does not suffer from collider bias because you're using genetically predicted rather than the actual measured phenotypes. And multivariable Mendelian randomization has been very useful in situations where there's clear pleiotropy, indeed because the processes might be so quite closely linked, like the triglycerides and HDL example, where the multivariable analysis always shows shows us a null effect of HDL. And because I'm in Harvard and Ben was involved in this paper, this didn't use multivariable analysis, but got the same answer, multivariable MR, which is the but you weren't doing IV estimation, so it's not, it doesn't count. So get similar. It does get similar answers, which is that the HDL cholesterol attenuates as you adjust for the other phenotypes. But in multivariable MR, you're doing it's a phenotypic effect, and in multivariable MR, your assumption of gene environment equivalence has to be is conditional on the other phenotype. So it's conditional. It's conditional equivalence of the genetic effect on the environmental effect. Some interesting ways forward in Mendelian randomization, for example, through fractionating instruments, through their different tissue specific profile. This is with the 900 BMI variants, looking at the ones which co-localize with the BMI signal co-localizes with an eQTL in brain, or co-localizes with an eQTL in adipose tissue, subcutaneous adipose tissue, or both, and then using the posterior probabilities of co-localization as the parameter, you can carry out multivariable MR. And the interesting thing is that exactly the same difference in body mass index, you get very different predicted causal effects for these two different sets of variables. So BMI influences all these things. This is the overall BMI, MR, but this is then using the two different sets of instruments, brain in red, which is seen to have these adverse predicted effects on these conditions, and much less so for the adipose tissue. And you think, well, gene environment equivalence should suggest you don't see this, but here, if you then take those genetic variants and then you look at how adiposity develops across life in Elsbach, for example, from birth on, you get at the same level of BMI, you have very different adiposity distribution in terms of peripheral vascular, in terms of visceral adiposity, etc. So this you actually have got gene environment equivalence. It's just the genes let you know what the true phenotype is. Because you have complex phenotypes like body mass index, of course, it is made up of many different components. So it's a great way of being able to unpack that. So MR with increasing GWAS size and increasing complex methodologies, these are definitely two-edged swords. The increasing GWAS sizes, of course, as the GWASs get bigger and bigger and bigger, you just pick up heritable confounders. So for C-reactive protein, for example, one of the top hits is FTO, the genetic variant that relates to BMI, the top BMI variant, because BMI increases your CRP. And genetic variants related to smoking and education, etc., start coming in. And I showed you this picture with this cis variant, beautifully unrelated. This is now if you use the polygenic score. So this is equivalent to doing a standard instrumental variable analysis, doing an inverse variance weighted IV. You've got really quite substantial confounding you're introducing. So you're often better off just using the cis variant. Here's a complex method, is using a nonlinear MR method which demendelizes the association within strata of the exposure, and that demendelization would appear to remove some obvious collider bias. Here are the observational associations that were observed, and you see the same U-shaped curves for all-cause mortality and for CVD, cancer and non-CVD. The low point where the line is crossed is about the median level of vitamin D in UK Biobank. And now here's the MR estimates for all-cause mortality overall. And what you see is that in the lower strata vitamin D, it appears vitamin D is really strongly protective. And it's protective in the other strata, but it's not protected overall. So you think, that's a bit odd. then I guess in the back of your mind you might think, oh, Simpson's paradox. I've heard of Simpson's paradox. You can see that you cannot see Simpson's paradox in this situation when you have a causal interpretation of the exposure and the outcome, and you know the distribution of the categories. This is just literally impossible. They showed the same thing when you see exactly the same effects for everything, and you think you've got confounding, and there is violation of some of the additional assumptions that are required. There's no free lunch with complex methods. Every method comes with a new pack of assumptions. One of the assumptions will be the linearity of the genetic variant intermediate phenotype, which often doesn't exist. It very obviously doesn't exist in the BMI situation, for example. So these results were spurious, and being on vitamins is particularly saddening, seeing as the crap epidemiology on vitamins is what drove us to work really on Mendelian randomization. Now it's been saved, and here's the authors introduced a new method, and they showed their original method. They show is biased. Yeah, so they agree that this is biased, but they say that their doubly ranked method is showing less bias. I talked about negative controls, which I think are massively underused. So one thing that that vitamin D can't do is change your biological sex or change your age. It might change how you feel, but it's not going to actually change your chronological age. So we just carried out using these two methods. We looked at, we did nonlinear MR, and it's amazing. Vitamin D just makes you old. Vitamin D deficiency makes you older, and it made really quite substantially. And indeed, it also changes your sex. So negative control is a really powerful way of getting a sanity check. I think there's a huge opportunities with a historically informed view of genetics and phenotypic causation. Here's T.H. Morgan in 1917. It might perhaps not be a very great exaggeration to say every gene in the germplasm affects every part of the body, or in other words, that the whole germplasm is instrumental in producing each and every part of the body. I can't remember if it was 1917 or it was when the omnigenic model was published in 2017, but the omnigenic model setup is basically a multivariable MR analysis of genes, of the core genes which are regulated, influenced by others. And natural selection can be studied by Mendelian randomization. In case control genetics association studies, the genotypes, the genes are dragged into the case group by the disease, and the evolution, the selection by phenotype drags along the associated genes. Gene environment equivalence, by the way, is assumed by the Breeder's equation. I won't go into that. It's quite, no, it's not. I don't think that's been much written about. And people are beginning to use MR to look at what are actually the things that are being selected for on what things are pleiotropic, because of course natural selection suffers from pleiotropy. In the famous artificial experiments with the farm fox, of course, the tame foxes got furry tails, which weren't being selected for, and people in the evolutionary field are beginning to use these sorts of models. Sewell Wright said, from a more general standpoint, all of this is merely an elaboration in terms of modern genetics of the conception of evolution by natural selection advanced by Darwin in the Origins of Species 100 years ago. Wright certainly saw that. So conclusions. Part one, Mendelian randomization can be useful. The second thing, rather sadly, is that there are an enormous number of poor Mendelian randomization studies. These are just two that I just got the latest two that go into my recommended for you. But this is assessing the role of lipid-lowering therapy using MR. But what they're doing is they GWAS which statin you're on. Are you on atorvastatin and simvastatin? And then they use those as instruments for using simvastatin and atorvastatin. And I mean, this is of course ridiculous. There's no way many genetic variants that will influence which statin you end up taking. It'll be your seriousness of disease risk, and education education, etc., etc. So of course you can do it. And here's causal effects of COVID-19 on cancer risk, where the studies they're looking at the outcomes in were collected when there was no COVID-19 around. So you can't be saying about the effects of COVID-19. You can say effects of liability to COVID-19, but that's a very different thing. We're to blame for this. These are the sort of citations for the 2003 paper. And I looked at this, so it was a very slow burn. Really, it was GWAS studies which started, people could do it. This is the year MR base was released, which allowed people to do 2-sample MR at home. We had a paper that was provisionally accepted by Nature Genetics called MR of Everything versus Everything, when we put everything into, which we got nice reviews for. Chip Amani led it, and we never resubmitted and got it published, because and in it we said we have done every single 2-sample MR analysis anyone can do. They can't be published because we've done them. They're now released. So you've got to bring something additional, some additional data, some additional biological understanding to make an MR study worth publishing. The value of MR depends on the data. We're lacking clinical cohorts to be able to examine disease progression. Tissue-specific data, I think, and precise macro-level phenotyping are key. And deep biological understanding is required. My conclusions, part 2, is that natural selection is a subcategory of Mendelian randomization. So, although that's not a joking bit, but Mendelian randomness is what allowed for the development of complex life. When people say, "Oh, it's not random," of course they are cheaters and everything, but the whole point is that the genetic cheaters are constrained by the randomization process of the Mendelian rules, and because otherwise you would just get genetic variants that just manage to cheat by moving to parthenogenicity, or which would lead to a short-term advantage, but long-term disappearance. So the existence of complex life depends upon those laws. And we have evolved to drink warm Budweiser. So that's the end of the conclusions. And that's, when you want to buy someone a Christmas present, that's the ideal Christmas present, honestly. Thanks very much.
Questions for George?
Right. So that was wonderful, George, and I love the historical perspectives too. But can you comment a bit about the increasing the precision of the exercise by improving the measurement of the exposure over a lifetime? Many of these Mendelian randomizations are predicated on a single measure of an exposure that may not represent the lifetime, the lifetime effect of that gene on that exposure. And, and how do we solve that problem?
Yeah. So I think that's absolutely right. I think each genetic variant, what you're estimating the effect of, is the effect of, you've got exposure of interest, and if you had a lifetime trajectory of how that exposure relates to the genetic difference over life, it might change over life. What you're estimating is the effect of that liability trajectory. And of course, for different genetic variants, the liability trajectories will be different. To look at the effect of lifetime, for example, when we did CRP studies early on, we genotyped kids aged 6 and showed that the variant had the same relative effect on CRP at age 6 as it did at age 50. So you can get some notion about how the genetic variants, what they are actually proxying. I think very much you need to have measures. You need to have measures across life for those variants. I mean, I think at the other end, thinking about what phenotype the genetic variant truly relates to is important. I mean, it's very clear that genetic variants that relate to BMI are relating to very different processes that are influencing BMI, and are relating to different subtypes of BMI, different classifications of BMI. I mean, the great thing about the genetic variation, though, is that your genetic variation will be within the bounds of biological normality when you're using when you're using genetic variants taken from just general population, common variants from general populations. So they will relate to biological processes which have some, have some sort of universality. So some of the sort of arguments about, using measures like body mass index, like saying, you could reduce someone's body mass index by cutting their leg off, but that won't make you live longer. There aren't any genetic variants for having your leg cut off. So what's nice about MR is that it actually does take you into something which is relating to the biological processes. But I think there needs to be a much greater understanding of precisely what the genetic variants are doing. So I think that's why the sort of tissue-specific data are so important. So, an example, I could have, I took out 20 slides. It would have made it 196. For example, I was giving that. I talked to some people at Biogen on Tuesday about with sclerostin. Of course, what people measure in studies is circulating sclerostin in the blood, but sclerostin isn't made to be something which is in the blood. It's generated by osteocytes in bone, and what gets into blood is leaked. Most of it, at least, and what gets into blood is just leaked out, and so you can have a variant that relates to higher production of sclerostin in the bone, and that's probably the cis variants in SOST, which have a much larger effect for a given level of sclerostin in the blood. They have a much larger effect on bone mineral density, fracture risk, etc., 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 because what's likely is that they are related to the actual osteocytic production, the actual production of sclerostin, whereas in the blood, the things which will influence sclerostin level in the blood will be everything from kidney clearance to some things which influence leakage, etc., etc. So we've really got to think about ways of getting that information, but you don't need to get that information on a million people. The great thing about using genes as these causal anchors and these links between different data sets is you could just have a few hundred cases of the really detailed, incredible phenotyping, but then you can take the genes to actually be able to estimate the effect of that sort of precise phenotypic difference on disease. That's why it's magic.
So I guess you could also argue that that some of the relationship between the instrument and the variable would be better if you had multiple measures. If you had multiple measures over the lifespan, and you might say that even the associations are underestimated because you've
So the thing with the instrumental variables is that in econometrics, instrumental variables was used for measurement error correction. Economists would predict, and it does actually. It does actually correct for measurement error, but it assumes some sort of randomness of the measurement error. But you will often find, as you do, for example, with the LDL cholesterol example, you find that the MR estimates are larger than observational estimates because they are corrected for measurement error. Right. But the measurement error model would be one of the one assumption
that I have concerns over is the sustainability of the liability in all situations. Is it equal and sustainable, that liability to higher BMI or whatever? I mean, we're in a situation where we've never had people weigh 600 pounds before,
no.
Ever in human history. And is that liability the same at these extreme ends?
So I think these are again beautiful things that genetics can look at, especially in populations which have really rich data, in particular historical data. So in Norway, you can go back and look at how the sort of polygenic risk prediction for BMI, what it looked like in 1960, and the slope is a bit. The absolute level of BMI, of course, was much lower. But basically, the variance related in pretty similar in pretty similar way.
If you do that with smoking, do you get the same thing?
I haven't looked.
I bet not.
No, you'd imagine.
Because I think there's bigger cultural shifts in smoking behavior over that time frame.
And also,
Anyway, more questions from the audience.
I mean, I think the thought experiment example of that I like is thinking about education scores in rural India, which will probably predict higher body mass index because you get higher income, you can actually eat more. Whereas in rich, high-income countries, the opposite is the case.
I've become more interested in looking at MR for drug targets that are kind of known to be causal for CHD. So, you know, if you look at the region for ACE, and there's actually nice PQTLs for the ACE region, there's nice EQTLs, but then there's kind of a null, there's, you know, null in the CHD, and in for a GWAS to CHD. Like if you look at SGLT2 region, there's nice EQTLs, there's even some variants for diabetes, and then it's null for kind of CHD and heart failure. And you could see a paper coming out now of like SGLT2 inhibitors are unlikely. An MR paper, if SGLT2s were developed in kind of the MR era, it's unlikely to have any benefit on heart failure or chronic kidney disease or CHD. So just your opinion on
Yeah. So I don't know those. I'd be interested to see those specific examples. So maybe you could give me some references. I mean, it's the GLP-1 receptor target, which in the MR studies doesn't relate strongly to BMI, but the drugs clearly have a substantial effect. And that is, I think that's something which really needs looking into, and you'd learn something really interesting by getting the answer. By getting the answer to that. I mean, one possibility is some form of canalization, but the thing I was saying about, it's about setting, about the tissue responsivity being set, but being differentially sort of set, so you get some sort of stabilization of the genetic effects, but you don't get the stabilization of, obviously, the stabilization doesn't protect you from getting a big whack of an agonist or antagonist. So, but I'm trying to put together these examples to look into them because of my obsession with canalization. So I'd be really interested to hear those.
Last question. Oh, we got one we'll have one from online, and then we'll have you as last.
So a question from the folks watching the stream online. Given your mention near the end of the kind of large number of poor quality MR papers out there, just a question of how wary should we be on average when we see just a Mendelian randomization paper kind of at first blush?
Yeah, very wary. I mean, MR, in some ways, it's gone like what happened with meta-analysis was that early on, the meta-analyses were really quite important, identified treatments which hadn't been thought to be beneficial, but when all the evidence was put together, et cetera, but then it just became a sort of paper mill type thing, because you can do them like you can do two sample MR, you can just do them without having to leave your bed. You can submit the paper before you have your first coffee. So extremely wary. I mean, the problem is the journals are don't know what to do. I get quite often contacted by journal editors, and they say, "What should we do?" I say what they do now is they should have a very simple screen, which is unless the submissions have come with a strobe checklist. So there's a checklist of what you need to report for an MR study to actually be able to evaluate an MR study. If they're submitted without that, it should be just rejected out of hand. I mean, it's amazing that some of these get through to publication, even in semi-predatory journals like the Frontiers journals are.
All right, last question.
Hi. Until now, I think that Mendelian randomization was primarily used for validating findings that actually had been already demonstrated in conventional observational studies, right? So now with the generation of all the large-scale omics data and the option that MR gives to use a two-sample setting, MR becomes like the first level of the first level of testing, right? So we are primarily using MR also for discovery of potential drug targets rather than confirming and validating potential association. So I just want your opinion on that, and how do you believe we should follow up on these findings, given that for many of those associations, we don't have the biological understanding that we have for associations constantly reported in previous observational studies?
I mean, I don't think MR validated a lot of observational analyses. You know, the ones I show, you know, the observational studies showed CRP was a strong predictor of coronary heart disease. Companies were starting to develop drugs to lower CRP. So the MR has tended to be the cold shower. When you know, unlike alcohol, absolutely really cold shower. And those are sort of situations when you had well understood variants like a cis variant in CRP promoter region or an alcohol metabolizing variant, etc., with negative controls. Yeah, so I mean that follow-up I think was very powerful. I take it so with the sort of screens, like you can have too many things to follow up, but I mean you're going to want to look at those for which you have the most the biological information which goes with supporting the instruments, supporting that you actually really understand the phenotype, because I think quite a lot of, I mean it's remarkable. I mean all the proteomics is extremely popular now. I mean, one of the things that I just always assumed you'd be able to go on for well-known proteins, you'd be able to do a quick Google, or probably could use ChatGPT or something for it to tell you about, you know, where it's produced, what regulates levels in the blood, etc. It's remarkably limited. For some of the proteins, it's just remarkable how little, you know, is known on those things, and then you're just using this sort of measure of something in the blood, and or in urine. And the interpretations really do depend on biology. So I think I don't think there's some rapid screen, sadly, that will take you to the rapture when, you know, some genetic finding is converted into a new drug that makes you a billionaire. I think that's sadly not going to happen.
Understanding mechanism is key, I think, is a good note to end on. So let's all thank George for this tour de force and celebrate him in the atrium.